Yeah, this company is Functional Circulation, and we make machines and methods that allow discovery of the organ. What that means is our machines can take isolated whole organs, and scientists, surgeons, can connect these organs to fluids, can control the temperature, the pressure, the various conditions, and then animate these organs to see how they will respond to certain stimuli. And they can do things with these organs in the machines that we make that they couldn't do in a whole body, and then as a result, learn new things that couldn't otherwise be learned. This is a demonstration of our first product, the Functional Circulation Model 30, and what it is a device that researchers, surgeon scientists primarily, can use to look at organs outside the body as a way to study them in ways that they haven't been able to study them before. And so what we have is a closed system that the organ goes in, and we provide oxygen, and we have a special fluid that's sort of physiologically balanced that is pumped through the artery of the kidney in this case. And so we connect tubing to the artery, and we also connect tubing to the ureter, which is the place where the urine comes out. And then we pump under controlled conditions, temperature and pressure, we pump that fluid through the kidney, and we observe what happens. And for the case of something like drug development, you might provide a drug into the artery and then see what happens, how it responds. Today, transplantation is facing older donors, sicker donors, and still looking for ways to transplant those organs. And so one thing you can do is to sort of take more donor organs and then check them out and see whether they're going to work right, and then only transplant those that you know will work right. And in a sort of a system like that, you could evaluate more organs and then transplant those additional ones that you weren't sure about in the past and maybe would have said no to, and then they go ahead and transplant them. I think in infectious disease, there's situations where certain cells that carry infection get sequestered in organs, and that they kind of don't know how to get them out. And so if you could then take these infected organs and then work on them in isolation, new areas might be developed that could address those diseases. I do work with university collaborators, professors across other various places in the country and around the world, in fact. One thing that we've learned, and this is a thing that happens with all businesses when they start, is they start to figure out what the market really can be. And so we've been looking now at rodent scale, equivalents of the machine that we make that you can test, like mouse hearts or mouse kidneys or that sort of thing. And it turns out that there's tens of thousands of researchers working, with genetically modified mice, for example. Yeah, the incubator is good. It's a place for me to sort of focus the work, of course. And right here is really nice too. I was happy that they had a little piece of lab space, and so I had a sink and a sort of this is kind of a more industrial setting here on Chicago Avenue, and it's fun, as well as the neighbors here are great. I'm surrounded by people who are doing really outstanding stuff, and so it's sort of like go over there and ask for a cup of sugar kind of thing. I certainly get that kind of stuff, as well as we just get to talk about science and technology and all that. So those are good things. I really got coming here because in order to apply for certain grants and to do certain work, you have to have a lab. And so, the incubator seemed like an ideal way to do it. This group has been really supportive. It's been fun to be here.